Thank you for attending today's webinar, DeepVariant to Improve and Extend, Use Training Not Programming, presented by Dr. Andrew Carroll. Dr. Carroll holds a Ph.D. in molecular biology from Stanford University and a bachelor's degree in physics from the University of Virginia. Andrew was Chief Scientific Officer at DNA Nexus, supporting many of the first large-scale genomics projects, such as the CHARGE Consortium and the St. Jude Pediatric Cancer Cloud. Dr. Carroll currently works as part of the genomics team in Google AI to develop methods for understanding genomic data and combining it with non-genomic biomedical data. Andrew shapes product strategy to drive adoption of the technology and ensure its connection to high-value application. His role has a strong focus on genomics community engagement, collaboration, and partnership. Without further ado, here's Dr. Andrew Carroll.
Thank you, and thanks everybody for joining this webinar. It's my honor to be able to tell you about some of the things that we're doing in Google Brain. Specifically, my talk is around how some of these new deep learning technologies that Google AI is developing allow us to rapidly improve and adapt technologies through finding training examples as opposed to writing new code. So as you're listening to this talk, there are three things that I want you to eventually take away from it. The first is this concept of deep learning, which you may have heard of as driving a number of the new innovations in AI. So the concept I want you to leave here with is that deep learning achieves high accuracy through modeling the unknown aspects of a problem. The second component I want you to take away is that as these deep learning methods are being developed, they begin to shift the importance away from writing very long and complex blocks of code to valuing more data and expertise about what are in those examples that are being trained on. So these two concepts together build the takeaway that if you want to rapidly improve and extend AI-based approaches, you can do so through finding and training on new data sets. So there's two sets of concepts that I'm going to need to introduce. The technology that I'm going to be talking about is one of the things that my team works on, a method called DeepVariant. And to introduce this fully, I will need to go through the process of variant calling in sequence analysis. So the process of understanding the data which rolls off of a sequencing instrument first involves taking the many short pieces of DNA which are produced by the machine and mapping those to a reference genome. After the reads are mapped to a reference genome, there's a process called variant calling, which identifies those positions which have statistical confidence to differ from a reference genome, as well as the genotype of the way that they differ. Once those variants have been produced, they can be used for a variety of downstream analyses, things like clinical report generation or cohort level understanding of populations. There's a huge diversity in the way that sequencers are run, and there's also many types of sequencers. There's an even further diversity in the types of downstream analyses that are performed, but there's not that many steps which perform the process of mapping and variant calling. And this is one of the areas that we decided to address in our development. So a very quick analogy for those who aren't familiar with the problem space of why variant calling is hard. The analogy that I like to use is that it's like taking a stack of newspapers all for a single day, blasting them into many tiny pieces, and then attempting to reconstruct them to say what is a given word in a given paragraph in this paper on a particular date. And in fact, the problem is even harder than this because newspapers probably have a very low typo rate. The machinery for sequencing actually generates small errors, and it's not necessarily clear which of these are errors and which of these reflect true differences between the sequences. So a key component of why this problem is hard is that our understanding of this problem is at least somewhat incomplete. What I've done here is I've tried to go through and enumerate the concepts that are required to correctly call variants, and I've tried to label them with my assessment of how well these are understood. So these range from things which are quite well understood to things which are very poorly understood, and there's a variety of things that we just don't know. So the unknowns of this problem are quite substantial. And they're also quite difficult to even get at experimentally. And as the field evolves quickly through new sequencing instruments, new library preps, basically as people are being more creative, the list of unknown unknowns for each different type of prep grows faster than we resolve these individual concepts. Okay, so this has been that's been a very high-level overview of the concepts of variant calling. I'm now going to briefly go through the concepts behind the work that we're doing in Google AI in deep learning. And to do this, I'll introduce 3 general concepts for the types of computer models that someone can apply. The first is this concept of statistical inference. I should clarify, these are my categorizations of the problem. You'll probably ask any other people, including many other people at Google, and get different answers and opinions. In statistical inference, there are a set of relatively fixed and understand mathematical transformations which answer particular questions about a problem. And these lead to digestible insights. And these are things like t-tests and things which generate p-values and such. The field of machine learning uses these statistical techniques to give computers the ability to fit parameters and weights to those in a way that's not specifically encoded, but is still largely based on the rules that have been written. So, an example of this is, I take a problem area and I find there are 6 or 7 things which are predictive for this problem, and then I use a machine learning approach to, for a given set of input data, fit the right parameters to build a classifier for those features which I have determined. So in machine learning, at least in this particular formulation of it, a key aspect of human knowledge is to figure out what the features are which are most predictive so that you can build models against it. In this new field of deep learning, which is starting to be developed, this is taken to one further raw level from machine learning. So instead of focusing on what the predictive features are going to be, the goal is to represent data in as close to its raw form and have larger, more complex models be tasked with the problem of understanding for themselves what the predictive features are. And so this pushes the expertise required to train this correctly to finding the data which is representative of your problem, understanding how to assess it extremely well, and building the architectures that will make this transformation possible to occur. So a deep neural network consists of a set of layers where connections between these layers represent mathematical transformations. The lowest layers of the neural network typically discover coarse or granular features. For image detection, for example, these often do image classification, these often do edge detection. While the higher levels of a network tend to learn more abstract principles, and this ultimately leads to a final classification. In this case, this is taken from this example is from ImageNet when you're trying to classify animals, for example, cat or dog. So in the work that our team has done, we have taken this image classification architecture and we have repurposed it for calling variation. And the way that we do this is we encode genomic information from a BAM file into a multi-dimensional pileup. You actually see examples of this over here on the left. These aren't quite the same sorts of pileups that you would see in a genome browser. They include multiple dimensions, things like base quality, mapping quality, some metadata about whether it supports the reference or alternate allele. So it's a little bit richer than a human would be able to look at. You also, it's not really an image in the sense that because it has this multidimensional component, it's really a tensor vector. This is fed into a model called Inception, which was designed to classify images, but can operate on this tensor data in a relatively generic way. And at the end, it produces a set of probability distributions for each examples. And for these 4 examples that are shown, these are the distributions that are generated on the right, which show whether or not the variant caller called this a reference, heterozygous event, or homozygous variant, and the confidence with which it made this call. So this work has been done over the course of several years. It was published last year in Nature Biotechnology after a relatively long development cycle. So we continue to develop DeepVariant for general purpose adoption and to support its application throughout the community. And these are the parameters that we value when we're thinking about how do we make DeepVariant better and what will the community care about. So the principal differentiating fact of DeepVariant is that it's the most accurate germline variant caller in a variety of external competitions and benchmarks. Furthermore, we value its robustness, that its accuracy advantage will increase on several hard data sets, and I'll give examples of this in a bit. We try to make DeepVariant as fast as possible, recognizing the community values this. For a single large machine, it will run in 4.5 hours. Try to make it as cheap as possible. So we use our benchmarks on Google Cloud, although you can run it anywhere. In the case study, we have 50-fold coverage whole genome. It costs between $2 and $3. DeepVariant is extensible. This is actually going to be a part of the talk later on. You are able to retrain for new technologies without the need to write new software. And finally, we value open source and the use of standard file formats. So we want to make DeepVariant as unconstrained as possible. So you can run it in any environment. You can check out the source for yourself. It's under permissive open source license. You'll be able to make modifications. And we're not using any sort of proprietary formats. We're taking information in BAM and CRAM. We're producing outputs in VCF and GVCF format. As part of making DeepVariant fast, I'll go through a number of the components behind this before I get into the main scientific areas of the talk. As part of making DeepVariant fast, it plugs into a variety of methods for acceleration. So this leverages the fact that DeepVariant is built on top of TensorFlow. TensorFlow is a library for deep learning, which is open source and developed by Google Brain. What you're seeing in this graph from DeepVariant 0.6 to 0.7, we enabled a variety of different accelerations. So you see the DeepVariant 0.6 and 0.7 is using AVX acceleration on a CPU platform. You can see we dramatically decrease the time required for the call variant step. We also enabled a special hardware accelerator called a TPU, which is designed to make TensorFlow as fast as possible. You see the further improvement in speed. And you can also get an improvement in speed on GPUs as well for a similar reason. We're actually quite happy that third parties have started to build their own acceleration processes on top of DeepVariant. So two different solutions, Parabricks and AT Genomics, have adapted DeepVariant in various ways for further acceleration. So Parabricks has converted all processes to be compatible with a GPU, and they have an accelerated framework for executing on GPUs. They also have done this for the mapping step. So they're able to run from a FASTQ file, map and run DeepVariant in 70 minutes, which is just about as fast as we've seen people be able to analyze a whole genome. And AT Genomics has optimized for Spark clusters. So this uses distributed CPU processing to be able to process in only a couple hours. All right, so now I'm going to get into some of the scientific stories for DeepVariant, and I'm hoping to hit some of the concepts that we went through at the start of the talk. So the first has to do what's behind the high accuracy of DeepVariant. And the thing that I want to propose is that part of this is the ability to model the unknown aspects of a problem. So the first thing I should probably show is just to demonstrate that DeepVariant is in fact accurate. So DeepVariant was the winner of the SNP Accuracy Challenge in the 2016 Precision FDA Award. Amongst the benchmarks I can show you, here is an analysis that was conducted by DNA Nexus, which compares a couple earlier versions of DeepVariant against some of the existing standards. And the difference in the reduction in false positives and false negatives is something that we see fairly consistently across a variety of samples and a variety of different benchmarks. This is using the HG002 PCR-free from Precision FDA, downsampled at 35-fold coverage. And I should point out that DeepVariant is never trained on this sample, so it's never seen any of these examples in the process of generating the model. So you can see not only do we have substantially better accuracy, also we've continued to improve this accuracy over time. We're now at version 0.8 and have continued to add functionality and improve accuracy with each release. Another component of the accuracy is that DeepVariant's understanding of its errors is better calibrated with reality. So what this plot shows is a comparison between the self-reported quality score of a variant and the observed quality score, which you can conduct in these benchmarks. You basically see for all the calls that are right and are wrong, what's the probability that they were correct relative to the true benchmark. So as the estimated quality, self-reported quality by the caller goes up, you see that DeepVariant's relationship between its self-reported quality and its observed quality is much closer to the calibration line than some of the state-of-the-art standard methods like GATK. And I think one of the reasons for this is that GATK has a fairly comprehensive and complex set of heuristics, which attempt to model all the different errors and the things that can co-vary between each other in order to figure out whether a position is variant or non-variant. But it's not able to kind of go beyond the features in the code that have been thought of by humans in the course of writing these methods. And DeepVariant is able to, within its neural networks, establish relationships that maybe humans haven't thought of, or potentially complex relationships between these features. For example, in a homopolymer C sequence length 5, base quality tends to decrease slightly relative to true expectation on the reverse read. This is not a real example, but that sort of relationship, if it exists, is something that can be modeled in deep neural networks, kind of intrinsic to the training process. So one aspect of this is that when sequencing data is noisy, the performance of DeepVariant seems to hold up reasonably well. So I need to walk through this example. This is a method called read shift, which was blogged about by DNA Nexus. And what the method essentially does is it leverages the fact that if you have a set of 350-fold coverage sequencing for a sample, you're able to selectively resample that in a way which can somewhat shift the read quality. So for example, you could go as extreme as, on the basis of the quality scores for the reads from this 350-fold coverage sample, give me the worst 30-fold coverage from that entire set. This is what it's trying to show in a kind of conceptual manner. So you can apply these shifts where you shift the overall quality distribution of a 30-fold genome from higher coverage. And that's just what you see on the right, unshifted, a control, which applies the shift machinery with no shift at all, and increasing shifts. And what you see is DeepVariant tends to be relatively robust to lower quality sequencing that has this nature. Okay, so the second story I'm going to go through is that in order to improve for novel technologies, these deep learning methods will allow you to quickly improve through training on new examples. Okay, so how does training DeepVariant work? Actually, training is very similar to the process of actually calling a variant, but with one exception. So you give it a set of labels. You give DeepVariant a set of labels which indicate what this position is that are associated with this example. And you train these in batches. We have, I think, 500 million different training examples for our whole genome models. And this updates the weights of each of these kind of individual connections between the layers. And this is done in batches, and the accuracy is measured on a separate tuning set until essentially the tuning reaches its highest accuracy and the model only starts overfitting as opposed to learning new things. And then you take that peak accuracy model on the tune set. So this can really just be applied from a set of examples generated from, say, a BAM file, and a set of labels from, say, the genome in a bottle for HD001 or HD005. So one example of this is the improvement that we see from DeepVariant version 0.5 and DeepVariant version 0.6. So when DeepVariant was first released, there were a few samples that people reported which didn't have that improvement in accuracy performance that I indicated for the prior PCR-free samples. And after a couple of them, we realized that the reason that these are not performing as well is that DeepVariant has only ever been trained on PCR-free data. So DeepVariant hasn't been able to learn this concept of PCR and the errors that are associated with PCR. So in order to improve this, instead of having to write any new code or new heuristics, we just went out and we got some examples of HG001 and 5, which are PCR positive, and then we're able to train DeepVariant with those examples. And this led to a dramatic improvement in overall accuracy for the PCR positive set. So I'm now going to divide this into two deeper investigations, one of which was the process of extending DeepVariant for a new data type, which is PacBio circular consensus reads. So as a very quick overview for circular consensus sequencing, so Pacific Biosciences is a sequencing instrument company which can produce very long reads from a single molecule measurement of DNA. And this has the advantage that long reads can more easily be mapped to the genome or assembled. And therefore, you can reduce the number of the errors that are associated with mapping, and you can extend analysis into regions of the genome which have been historically much more difficult to analyze correctly. So circular consensus sequencing is a way to get the benefit of both long reads and high accuracy. So in this, a circle of DNA is constructed, and a polymerase reads that same circle, or copies from that same circle multiple times, and each of these runs around the circle is aligned to each other, and this produces a consensus. So this is a sequence which is both long and of quite high overall accuracy. The work that I'm going to present was published, it's in a bioRxiv preprint in early January, where we kind of extensively have analyzed the CCS technology and the retraining of DeepVariant. So if you're curious, please read this manuscript for more details. So when we're adapting DeepVariant for the PacBio CCS reads, we look at what's different about this technology relative to the standard Illumina technology. And so for the overall SNP accuracy, the difference between PacBio CCS and Illumina is not particularly significant. So here you see the accuracy for applying DeepVariant with the PacBio circular consensus sequencing model that we train, and GATK with a couple different filtering criteria. These numbers are pretty similar to the numbers that you would see from Illumina sequencing. And also for the DeepVariant Illumina model, you also see numbers in this ballpark as well. I think that's between the GATK and the DeepVariant PacBio CCS model. So the SNPs are reasonably similar. It's definitely possible to do better by training on them. But the biggest impact is actually in indels. So the error profile for PacBio CCS reads is just different from Illumina sequencing. They're much more likely to have indel errors than they are to have SNP errors overall. So because this data type looks so different, the challenge in calling from it shifts from calling the SNPs correctly to really calling the indels correctly. And here's where you see a quite substantial difference between the software that's currently been written and what's possible to achieve. So even with the hard filters applied to GATK4, the best accuracy that was able to be achieved was around 80% for the small indel accuracy. So through training on this PacBio CCS, we are able to take DeepVariant up to 95% accuracy, and we've since, in the release version on our 30-fold case study, we have 98% indel accuracy. So just through the process of finding examples with only one tiny code change, we basically have a reassembly process for short reads, which built a De Bruijn graph onto candidate haplotypes. We disabled that for PacBio because the PacBio reads are so much longer than the haplotypes that we would have made by short read assembly anyway. With only that code change and then finding examples for the CCS data, we're able to dramatically increase the accuracy to make this competitive with other Illumina-based technology, even for indels, which has historically been the difficult thing for long-read sequencing. So what you're able to see on the right side of this figure is that with this technology and with this model, we're able to have methods which we'll comprehensively call small variants, SNPs, indels, and structural variants in a single sequencing technology, which has been quite exciting. This is something that I alluded to, which is that as you have more data for models, you will often be able to learn deeper associations and to overall improve accuracy. So, when we did that work, we only had 28-fold coverage from a single sample and from only one of the types of PacBio chips. From this publication, we have, over the course of the last couple months, prepared production-level models for this, which we have released in version 0.8 of DeepVariant. And with the 100-fold coverage PacBio CCS, subsampling that into a variety of different coverages from a variety of different runs and from different chips, we're able to learn more general aspects about the problem and further improve the overall accuracy to 98% in our 30-fold case study. It isn't just a question of having more data. One of the key things in the process of training is to figure out how to make your training sets as broad, diverse, and representative. In general, the more different stuff you can throw at a training regime, the better the model will be able to generalize. So an example of this is we want to perform well for all of the different coverages that you could run your PacBio CCS at. So we take that 100-fold coverage. We do train one full 100x run, so we want to be good for things like targeted sequencing that does go to very high depth. But we do most of our training by downsampling this from 9-fold coverage to 30-fold coverage. And what this allows us to do is to be robust against a very wide range of coverages. And so you can see here that accuracy holds up quite well on the DeepVariant CCS models for coverages of 15-fold and even, especially for SNPs, lower. Oh, sorry, I skipped the slide. So the second part is going along with a similar theme, but instead of looking at new sequencing technologies, we're going to be talking about adapting DeepVariant for non-human genomes. So when we built DeepVariant, we wanted to understand how does its performance generalize to things that aren't humans. One of the first collaborations that we conducted was with the International Rice Research Institute, which has the 3,000 Rice Genomes project. And we leveraged the fact that within these 3,000 rice genomes, there are a number of inbred strains. And these inbred strains have a principle that a heterozygous call is probably wrong because they've been quite depleted of variation in the process of generating that inbred strain. So you can model from expected heterozygosity exactly how many het calls you might expect for a small number of remaining heterozygous variants, and you can run freebase and GATK. In this case, DeepVariant is only the human model of DeepVariant. It was never retrained for this rice data at all. And so what you can see here is over the course of the rice chromosomes, the homozygous calls and the heterozygous calls that are colored for DeepVariant and for freebase. So you can see freebase and DeepVariant will both make homozygous calls, but the vast majority of the heterozygous calls are made by freebase, which doesn't match our biological intuition for this problem. So as I said, you can infer the error rate for this relatively challenging data set because these are low coverage rice genomes. And you can see that DeepVariant substantially outperforms the accuracy of some existing methods. So DeepVariant performs very well on these strains of rice. Another investigation that we conducted was to see how DeepVariant will perform for mosquito genomes, and this was done in collaboration with the Sanger Institute around a project called MalariaGen, which seeks to understand how the genomic relationships between humans, Plasmodium, and mosquitoes relate to each other to help control malaria. So one aspect of these malaria genomes is that the mosquito genomes actually look very different from human genomes or from inbred rice strains. Most specifically, mosquitoes, as well as a variety of other insects and of course many other plants and non-human organisms, have very fast generation times, and they also have large population sizes that have been interbreeding over substantial periods of time. So they have a substantially more genetic variation than is present in humans. This is just showing a window of these father-mother-child pedigree. And just within here, there's 4 variants in 40 positions, which is 2 orders of magnitude greater than the variation rate in humans. So the reason I show this is that when we applied DeepVariant to mosquitoes, we noticed that DeepVariant would often prefer not to call some of these events which we think actually look reasonably reliable. I think that in this case, in the mother, this green G to A variant was not called. And so we noticed the behavior started to depart from what we would expect as ideal in these mosquito genomes. So what we did is we took the pedigree information. We had, I think, 11 children and 2 parents, and we divided this pedigree. So we would not evaluate on 5 of the children, and we would do the training only on the child, and we would find positions where we could confidently identify the child's genotype from a confident call in both of those parents. And then we're able to generate both homozygous reference and heterozygous positions because we can find places where the mom and dad are homozygous different or any combinations of this. So from this, we make a set of examples that we can train in a very similar way to the way that we do for Genome in a Bottle on these very well-characterized human examples, where we have for this child a set of positions and then a label associated with it. So when we do this, we're able to dramatically improve performance on the mosquito model. So what you see here is the DeepVariant version 0.7 human model, GATK4, and DeepVariant 0.7 retrained mosquito model. These are the variants called with genotype quality greater than 0, meaning any variant which is called. And these are the variants that are called with genotype quality greater than a Phred score of 20, which means these are the high confidence variants. So in this retraining process, we're essentially able to build mosquito-specific, or maybe it should be said, variant-dense organism-specific model, which substantially outperforms state-of-the-art. And again, this calls back to the plot where I showed the calibration between observed and empirical quality, that not only are the calls better with respect to Mendelian violation rate, it's also the case that the understanding of why something is likely to be wrong is better as well, meaning those calls which are made with high confidence are very, very unlikely to be errors, while state-of-the-art is limited based on the quality of that calibration curve, which I indicated earlier. I will comment one last thing on why, or basically what is the difference between human genomes and mosquito genomes. I mean, what has DeepVariant learned for these variant-depleted genomes versus what it has learned now in the mosquito-dense model. It's not necessarily possible to comprehensively say, but I think a very likely explanation, we have some other evidence that this is the case, is that there's this signature from segmental duplication, where a region of the genome is copied to a different part of the genome somewhere else in the evolutionary history of an organism, which sometimes it's incompletely assembled, in the reference, meaning that segmental duplication isn't represented. And sometimes reads between the two mismap, and when they mismap, they carry this signature for variation to other parts of the genome. So this makes a signature which looks like variance, but it isn't actually variance. And in humans, this is often characterized by a set of variant-dense heterozygous, or things which appear to be heterozygous variants that are densely packed together, reflecting the divergence of that segmental duplication. And we think that it's likely that DeepVariant has learned to try to protect itself from making errors by calling segmental duplications. And in humans, the fact that humans are variant poor or variant depleted means that it can be more aggressive at doing so. Whereas in mosquitoes, there's so much variance that a variant-dense signature is more likely to be true variation. So we think that between the mosquito model and the human model, DeepVariant is essentially learning different sets of parameters that reflect the evolutionary history of these organisms. So I've shown you a variety of these different examples. One of the key points that I want to make is that in certain of these examples, the out-of-the-box DeepVariant model performed best, and in certain of these examples, the DeepVariant didn't perform as well as an out-of-the-box method. So this is actually a general principle for everything. Every problem is different, and when you're considering what tools to apply to every problem, you always need to look at whether your methods generalize. And this is true for code that you've written, it's true for traditional machine learning approaches that you've used, and it's true for deep learning approaches. The point of this section of improve by training, not through writing code, is that when you find cases where your deep learning method doesn't generalize as well, and of course you should always be looking through benchmarking, you can very quickly improve this if you're able to find good examples and good labels and train to adapt the method to the new problem class. So finally, I wanted to acknowledge all the members of our team. DeepVariant isn't the only thing that we do. I hope to be able to talk about many other future exciting projects. The folks on this team have a combination of expertise in both genomics and deep learning and software infrastructure. So it isn't the case that we have a bunch of just machine learning and deep learning people who are trying to hit everything with a hammer. We definitely value this combination of expertise, which we feel is essential to being able to generate good methods in this space. And thank you everyone for listening, and I'll be happy to take questions now.
Thanks, Andrew. That was really great. We're now going to move to the Q&A portion of this webinar. We had a few questions come in. The first one is, can DeepVariant work with tumor cells? Does it do tumor only or tumor normal calling?
Yeah, so the methods that I've presented here are for germline DeepVariant. So DeepVariant has been trained specifically around identifying germline homozygous heterozygous variant positions. We have an adaptation of DeepVariant, which has been designed to call somatic samples, which is in early access mode. So we're reaching out to the community to figure out if there are people who can help us to benchmark and understand this somatic method. But DeepVariant itself is, the results I presented in the tool available in the DeepVariant GitHub are not designed for tumor calling. So they won't be able to detect subclonal variants particularly well.
What is involved in training DeepVariant? Is it possible for other users to train new models?
Yes, so we have tried to make it as straightforward as possible for users to train their own models. So if you go to our GitHub, we should have documentation which is available, which goes through a couple case studies that show how to retrain DeepVariant. One of these is an example for retraining DeepVariant on BGISEQ data, which should have a working line-for-line based example that you can at the end up with a DeepVariant model. There are a couple groups who we know have successfully gone through this exercise and have retrained DeepVariant on their own for different problem classes. But it is, it definitely is a process where you have to know the data set that you want to train on. You have to know, for example, have genome in a bottle samples. And it's definitely more computationally intense and difficult to get working than just running DeepVariant.
Next question is, what does DeepVariant work for out of the box, and what needs to be trained? Do you need to train for your machine, or need a different model for HiSeq-X or NovaSeq?
Right. So a very common question that we get is for these deep learning methods is that people don't have an intuition for, do I need to retrain on everything, or do I need to retrain on nothing? People tend to think they need to do more retraining than they do. So we have trained DeepVariant on a mix of HiSeq 2500, HiSeq-X, HiSeq 4000, NovaSeq for both exomes and for whole genomes, and across a variety of different runs conducted at different institutions. So this general model seems to work pretty well for all of those different instance sequencing machines and types of runs. So in general, if you are doing sort of the most standard applications of sequencing with typical instruments, you don't need to retrain. You need to retrain if your problem is substantially different in the way that it represents or considers data. For example, the error model of CCS being quite different from Illumina, or somatic calling being just different category. But for the most part, if you would run a method like Freebase, you should be able to run DeepVariant in a similar way without needing retraining.
Why is Google doing this? Are they trying to make money from DeepVariant?
Yeah, so this is also another very common question. So DeepVariant is open source. It can be run anywhere. It can be modified. We're not charging a license to it. There are a few aspects of DeepVariant which make it run better on Google Cloud, but this is not by like our specific design. It's really the fact that Google has a couple accelerators for TensorFlow, which is the engine behind DeepVariant. In any case, the goal of DeepVariant is not specifically to drive Google Cloud adoption in any case. The reason that Google has put so much effort into TensorFlow and into methods like DeepVariant is that deep learning is a critical part of Google's overall technology stack. It enables very interesting features. For example, like on the Pixel phone, when you take a picture in low light, you get a kind of a better picture for that condition, and for translation, you can have English Chinese, Chinese to English more effectively done. All those technologies use deep learning very substantially under the hood. So realizing that this technology is so essential to the way Google operates, Google has tried to be very open and democratic with the libraries that it has built in TensorFlow. And I think one of the key things for this is that we at Google always want to ensure that our technology is running on the best underlying stack. And the best way to ensure that that's the case is to make sure that stack can be as broadly adopted as possible. And DeepVariant is an example which further proves the value and usefulness of TensorFlow. And this allows TensorFlow's further adoption throughout the community. And I think this is why it can be very valuable for Google to produce a method like DeepVariant, even if there isn't a direct revenue expectation from it.
Well, it looks like in the interest of time, we'll have to stop here today. Once again, I'd like to thank Andrew Carroll for his presentation and all the attendees for joining us. If you'd like to watch this webinar again, the recording will be available at gmasweek.com. Also, check out gmasweek.com for more activities, including webinars, exclusive promotions, multiple chances to win a total of $20,000 credit for GMAS services, and much more.